Hello, in this video I'll take you to see what's inside the molecular biology lab. My lab is on the 4th floor. If you want to go to the lab, we need a student card to get inside. Just tap on the recognition machine and the door will open like this. And yeah, welcome to the biology lab. The first thing that I always do is turn on the light, air conditioner, and check around the place. Then also turn on the ice maker for DNA experiment, because when we are doing DNA experiment, our sample have to be in a cold temperature. And here we have gel electrophoresis. We use this step to separate mixture of DNA, RNA, or protein according to molecular size. And also we have pipette, tip, and for sure we need agarose for that. In this lab, we have also a lot of ethanol we use to preserve a sample. There is also thermal cycle machine for step PCR DNA, and for sure UV visualization to see the length of DNA fragment size of the sample. And we also have a dry machine. After we do sterilization tools, we use this machine to reduce a water concentration. We have to make sure the bacteria cannot grow anymore for our experiment. This is we use for move phenol or chloroform fluid because they are very toxic and dangerous for human. This is the view from window. Whenever I feel sleepy, I just go to this side. And here's also we have sterilization machine. So before we use a lot of tools here, we need to put inside the machine to make sure there is no bacteria anymore. And also have a pipette of various size, tips, and lots of chemicals. There is a buffer, DE, STS, salt, mineral oil for a PCR step experiment. These are samples from Indonesian island that I took. There is a mudskipper, fiddler crab, barnacle, and squid, while on the other side, the sample is collected from Bangladesh island. There is a centrifuge and vortex here. On the other side, also have a fridge containing a primer, DYE, for electrophoresis. We've got a lot of freezer unit in this lab. Here -20 degrees Celsius for DNA product, while 0 till 4 degrees Celsius, this is diluted DNA. These are we use for a PCR process. And here's some cleaning equipment in the lab. Usually every Friday I'll do a clean sweep of the room. Okay, that's it for DNA laboratory tour. Thank you for watching. Bye bye.